Hello, we are Group 17 and our presentation is on Werner syndrome. Werner syndrome, also known as adult progeria, is a rare disorder that is recognized by its symptom of rapid aging. Due to a mutation in the WRN gene that creates an ineffective and undersized Werner protein. The WRN gene is responsible for producing that Werner protein, and that protein maintains and repairs DNA, as well as assists in DNA replication. We hope that after our presentation, you will understand the following about the WRN gene: protein structure, gene structure, mutations, gene therapy, and more. The native function of the WRN gene encodes a RECU subfamily member of the DNA helicase protein, the Werner protein that aids in DNA repair. It is inherited through autosomal recessive inheritance, and it is expressed in the bone marrow, endometrium, liver, and all other major bodily tissues.
So for the WRN location and sequence, it is found on the short P arm of chromosome 8 between 12p and 11.2p, as seen in figure 1, and it covers approximately 140 kilobases and contains 38 exons. It is located above the centromere, and as you can see on figure 1, it is located between 12 and 11.2p.
Werner syndrome is characterized by the early onset of the appearance of features associated with normal aging and cancer predisposition. The first symptom seen with Werner syndrome, and it's often recognized retrospectively, is the lack of physical development during the early teen years, and specifically the lack of a significant growth spurt. Many symptoms appear in the early 20s. These symptoms often include loss and graying of hair, hoarseness, and scleroderma-like skin changes, bilateral ocular cataracts, type 2 diabetes, hypogonadism, etc. Most individuals are diagnosed with Werner syndrome in their late 30s to 40s. Mutation. Werner syndrome is caused by one of the over 60 known mutations in the WRN gene. A majority of these mutations result in a shortened, non-functional Werner protein. Research suggests that this shortened protein never gets transported into the cell's nucleus, where it normally interacts with DNA. As explained briefly earlier, the absence of a normal Werner protein in the nucleus results in disruption of DNA replication, repair, and transcription. It is not fully understood how mutations in the WRN gene lead to the signs and symptoms of Werner syndrome.
Werner syndrome is an autosomal recessive disorder, meaning that both parents must carry the recessive trait for their children to be affected. If a person is found to be infected, it means that the person's parents are both carriers and their siblings are also impacted. This is the case for all autosomal recessive genes. Parents who are both carriers have a 25% chance of having a child with Werner syndrome. Additionally, Werner syndrome is thought to be the result of a founder mutation and thus is related to the level of consanguinity in a community. Consanguinity arises from the marriage of two people who are related. As a result, Werner syndrome is observed at much higher rates in Japan and Sardinia, where the heterozygote frequencies are thought to be as high as 1 of 166 and 1 of 220, respectively. These are usually ethnic-specific mutations that are the result of the mutation occurring in a small population a very long time ago, and then growing in prevalence over time due to interfamilial marriages. This has happened most dramatically in Japan, where 75% of recognized Werner syndrome patients from 1904 to 2008 were of Japanese origin. As for the gene product, the Werner gene typically encodes a member of the RECQ subfamily of DNA helicase proteins, a class of enzymes that catalyze the unwinding of DNA base pairs during DNA synthesis. Encoded nuclear Werner protein maintains genome stability by playing an important role in DNA repair, replication, transcription, and telomere maintenance. More specifically, the Werner protein assists with the repair of double-strand breaks, which are potentially lethal DNA lesions generated by exogenous or external and endogenous or internal processes. Werner protein has 4 domains. First, there is the N-terminal 3' to 5' exonuclease domain, which catalyzes the hydrolysis of unpaired or mismatched nucleotides during DNA replication. This is otherwise known as a proofreading ability.
Next, there is the ATP-dependent helicase domain, which catalyzes the unwinding of double-stranded nucleic acids such as DNA. Then, there is the RECQ helicase conserved region, which helps bind the DNA substrate to initiate the unwinding process. Finally, there is the helicase RNA-ST C-terminal conserved region, which recruits the Warner protein to double-stranded DNA breaks. Below is an image of a normal RECQ-like helicase attached to its DNA substrate.
Unfortunately, because the Werner syndrome has broad effects across the human body, requiring gene therapies to target essentially all cells at the same time, and because the population affected is relatively small, there are no gene therapies currently under development. However, there is another alternative, which is somewhat viable, namely in vitro fertilization with genetic testing. Because the Werner syndrome is caused by a double recessive gene, it is entirely possible to detect it in parents, and if you know which parents are going to be at risk for this gene, you can then use IVF and embryo screening to determine which embryos receive the double recessive gene and terminate those embryos, ensuring that the embryos which are implanted are not positive for Werner syndrome. Hopefully, in the future, there will be advances made in technology such as CRISPR, which allow for retroactive treatment of individuals with this syndrome. But right now, there is no such a treatment on the horizon.